All right, guys, how's it going? Today we're going to get together talk a little bit about the cell cycle. Um, and this is some pretty crazy stuff here, so hopefully you guys hang with me on this. It's kind of a little bit longer notes as well, but we'll take some breaks throughout. I'm holding a baby, so if you hear some crying, sorry about that. It's the hazards of being home with a newborn and recording a video that you can't stop or pause. All right. So let's get cruising. Before we start, actually, one thing I want to say is I've noticed that a lot of you guys are not turning in work. It's going to catch up to you. All right. So if Ms. Hartsock asks for assignment or assignment is due, you need to make sure that it gets in the bin or gets to Ms. Hartsock. If not, you're going to accumulate a lot of zeros, and you know I don't take any late work. Okay. So basically, what I'm trying to say is get it together. All right. Turn in assignments when they are due, please. Check your grades online. I'll be grading, and if you got a zero because you're not turning anything in, then you got a zero. Okay. All right. So with that said and done, what I want to do is I want to review a little bit about what you guys saw yesterday with DNA replication notes, okay? So we know that basically DNA is going to be found inside of the nucleus. It exists as this double helix. And what it does is that basically it's in the nucleus. It's in a loose mass known as chromatin, okay? Think of it as like a bowl of pasta or just a bunch of yarn sprawled out all over the floor. It's very loose and it's relaxed, okay? The analogy, really bad analogy I like to give is like when you go home, you get off your school duds and you put on some comfy clothes, okay? So this is DNA in its home, the nucleus, in its comfy clothes, its loose state, okay? Now, what's going to happen is that it's going to get into a much more organized matter. And in order to get that started, it's got to wind around things known as histones. Histones are kind of similar to spools, where if you're spooling something up, you'll wrap it around it to make it a little bit easier. It's going to wind, it's going to wind, it's going to wind, it's going to wind, it's going to wind until it actually ends up being this chromosome. And if you guys check this out, look how squiggly that is. That is DNA that is wound up so very tight. DNA is huge, you guys. I think it's like 6 feet long in our tiny little microscopic nucleus. If you guys remember looking at the cheek cell, that little black dot, you have 6 feet of DNA inside of that nucleus right there. Okay, so some crazy things have got to happen. All right. Also, this right here is a chromosome. Okay, you guys wrote this on the back of your notes. Centromere is going to be this central part right here. Okay. What we call these two things, if you notice it's an X, we actually refer to this as a sister chromatids, okay? So here is one chromatid, here is the other chromatid, okay? These are two chromatids, we call them sisters. Think of them as like Siamese twins, they're attached right here at this centromere, okay? And in the cell cycle, we're actually going to do surgery to separate these sister chromatids, but they're identical copies of each other. Here was the original strand, here's a copy of it, okay? Anytime you see an X, I want you to think that DNA has been replicated or copied through the process that you guys learned about yesterday. Okay? So again, chromatin, loose mass, winds up into this chromosome. Whenever you see an X, one side of the X is the sister chromatids, one chromatid, the other chromatid, and here's the tricky part. This whole thing right here is a chromosome, okay? So this is a chromosome made up of two chromatids. If you were to actually look at a chromosome, I love this because you see all the DNA kind of like oozing out of this one. That's actually what it looks like, okay? All right, so with that, let's go ahead and move to our notes, okay? So you guys have these pictures right up here on your notes. I know that you guys don't have this right here, but you can put like a little brief little thing like centromere, chromatid, chromosomes. Doesn't really matter. You do whatever you need to do, okay? So let's do this first one together, all right? How many centromeres do we actually see on this one? Well, centromere is that central part. So what I see is I only have one, okay? How many chromatids? What kids often do is they count, that's one, that's two. We're going to count a chromatid as one great big huge line like so, okay? So that right there is going to be one chromatid, okay? How many chromosomes do I have? Kind of a rule of thumb here is how you can count your chromosomes is see how many centromeres you have, okay? Your centromere and your number of chromosomes will always be the same. So I have one centromere. This is also going to be a chromosome. Now some kids are like, well, Miss Chris, you called this a chromatid and a chromosome. Yeah, I did. And that's why it gets a little bit confusing sometimes. Okay. But we're going to practice a little bit more with that. Okay. Let's hop to this one, which you guys already saw the answers right there. Okay. So let's hop to C. And I know I skipped B, but that was on purpose. Okay. Let's do this one. How many centromeres do you see here? So how many central portions do you see? Hopefully you guys see one. Okay. How many chromatids do you have here? Now this is a little bit different, and actually this might be confusing at first, but once you get it, you got it. Okay, so right here, there's one chromatid, and right here, extends all the way through, there's the other chromatid. There's actually two of them. This is showing a replicated chromosome, the sister chromatids. So how many chromosomes do I have? Okay, remember my rule of thumb, count how many centromeres you have. There's only one centromere, so you're only going to have one chromosome. Okay? All right, what I want you guys to do right now is try B and D all on your own, okay? And Ms. Hartsock, you can hit pause on this video while they work on that. So try B and D on your own. And I'll leave this up here for you, okay? All right, so let's check these out. Again, you just saw the answers. All right, so this was B right here, okay? So how many centromeres do you guys see here? Right here, you're actually going to have two, okay? 1, 2. How many chromatids do you see? There's 1, and then there's 2, okay? So actually, you're going to see 2 chromatids. This is showing it a little bit different than a replicated chromosome, okay? But again, it's basically going to be the same thing, okay? All right, last number of chromosomes that you see here. Remember I told you to count how many centromeres you have, okay? So here I have 2 centromeres, here I have 1 chromosome, here I have 2 chromosomes, okay? So the magic number here is 2, 2, 2, okay? All right, let's hop to D. How many centromeres do you see here? You should see 1, 2 centromeres, okay? How many chromatids? 1, 2, 3, 4. So you're actually going to have 4. And how many chromosomes do I have here? I'm actually going to have 1, 2. Okay, you're going to have 2 chromosomes. Okay, so chromatids make up the chromosomes. Okay, all right. So let's talk a little bit about the cell cycle. All right, so what's going on here? Now, cell cycle is basically, to the purpose of it, is to grow, to replace old cells, or for reproduction. Okay, so we are growing nonstop, and this was actually some stuff that you wrote down for DNA replication. We're constantly replacing our old cells. Anytime you scratch your face, you guys, you actually are losing cells, which is really kind of gross. Your body has to replace those. And what I mean by reproduction is basically a type of asexual reproduction. We talked about this back in Characteristics of Life. This is where this guy right here is making more cells to produce this baby. And this baby right here is an identical copy of this one. Okay, so asexual reproduction is kind of creepy, but kind of cool at the same time. They are identical to each other. All right, so how does this happen? All right, now basically how we're going to make new cells is going to be kind of tricky. All right, we can't just chop our cells in half. The reason why we can't chop the existing cells in half is because everything that's in that original cell we need in the new cell. Okay, we need the DNA, we need the organelles, we need the cytoplasm, we need all of that. So the process in which we make these new cells is actually going to be kind of tricky. Okay, and it's a detailed process, okay? So the cell cycle is made up of 3 basic phases. The very first one is something called interphase. So in your notes where you see interphase, why don't you guys put a number 1 beside that? So put a number 1 beside interphase, please. Okay? All right, so what is going on in interphase, okay? The purpose of it is basically called cell preparation, all right? We're getting the cell ready to divide. All right, so please write down cell preparation, getting the cell ready to divide. You can't just cut this cell in half. You just can't, you can't, because like I said before, everything that's in this original cell, I need in the new one. So we gotta do some work, we gotta do some business here. Okay, so it's the cell preparation. Interphase is the longest stage. Okay, it takes a long time. There are some cells that spend days in interphase, and there's some that actually go through it a little bit faster, but still it takes a long time. Okay? All right, so interphase can be broken down into 3 specific phases. Okay? The very first one is something called the G1, or first growth, or growth 1. Okay? That's going to be the very first stage, so we're putting this under A. All right, this is basically where the cell says, hey, you know what? I'm getting a little bit too big for my britches. And believe it or not, it knows when it's getting a little bit too big and it knows when it needs to divide. Okay, it has hormones that help out with that. The cell's pretty cool. All right, so what it's going to start doing is that it says, all right, we got to start making more components that we have inside of here. We got to start making more organelles for the new cell. So if this cell right here has 20 mitochondria, it's got to whip up 20 more mitochondria. It's got to make cytoplasm. It's got to make another nucleus, okay? How does it do all that? The directions for all of that is actually in the DNA, okay? DNA is like a big old recipe book for what makes us who and what we are, not just our characteristics, but also what goes inside of our cells, okay? So first growth, the cell starts to make more organelles for the new cell. That is G1, okay? The second one, and I want you guys to put a star beside this. Everybody put a star beside B. The reason why I'm telling you to do that is because the EOC loves this one for some odd reason, okay? B is going to be synthesis. Synthesis basically means to make, or it's the S stage, okay? What's happening here is exactly, whoa, baby. What's happening here is actually what happened, what we talked about yesterday, okay? This is where DNA is going to be replicated. See, Madeline doesn't like synthesis either. She thinks it's ridiculous. So DNA is going to be replicated in the nucleus. So what we talked about yesterday, where you make an identical copy of that DNA, that's happening here during this stage, okay? S synthesis, okay? All right. The very last one that we have here is actually called G2, all right? And that's second growth or growth 2. This is basically where the cell wraps everything up, okay? If we need a couple more ribosomes, this is when it happens here. If we need to add, I don't know, another chloroplast, because this happens in plants as well, you can do it here as well, okay? So second growth is basically where they're making sure that they've done everything that they need to do and that we're ready to proceed on. Okay, so interphase again, we're making all the stuff to go in this new cell, excuse me, and we are replicating our DNA. We have done nothing to this cell, just basically doubled its contents. Okay, so interphase, we are doubling the contents of the cell. Okay. All right, now if you guys notice, you have this picture right here. We are actually, once we're done with this notes, you guys are going to go in and fill this in on your own, okay? And I have a little diagram to help you out with that. So we're just going to leave this blank for right now, okay? We'll talk about it at the very end. All right, so if we were to look at a cell in interphase, okay, but before we do that, let's take a look at this one right here. If I were to give you a picture, would you say that this is a plant or an animal cell? Pay attention. Hopefully everybody out there is thinking plant cell, correct? Okay. The reason why it's the plant cell is because it's square. Remember, plants have that cell wall. All right, if we were to look at an actual cell in interphase, it's really boring, okay? It's kind of like what you saw with your cheek cell, all right, where you have this, and then you got, there's your cell, you got the nucleus, and this one you can actually see the nucleolus, which is pretty cool, okay? We can't do that. Our microscopes are not that good. All right, plus your cheek cells are not necessarily undergoing mitosis, but that's a whole nother topic, okay? So that's a plant cell. In us, a little bit more boring, okay? Here's our cells, there's the nucleus, okay? So interphase just kind of looks like a normal cell. You can't see anything. And the reason why you can't, I won't get into that. Well, I will. The DNA is in that loose mass form. It's in its chill out form. It's in its house pants inside of that nucleus, so you can't see it, okay? So it's just going to look like a normal cell, nothing very exciting, okay? All right, so let's move on to our next stage, which is called mitosis, okay? Put a number 2 beside mitosis, please. Number 2 beside mitosis. Number 2, number 2. Okay? This is an animal cell, by the way. All right, so what is going on with mitosis? Well, what's going to happen here is this is where we're going to split apart that replicated DNA, okay? We're going to perform surgery on those sister chromatids, that X of a chromosome, and we're going to separate them, okay? So we're going to divide the DNA in the nucleus, all right? That's the whole point, all right? We're dividing the DNA, and actually when we divide that DNA, we're going to end up with 2 new cells, okay? So we're dividing the DNA, and you can see the steps right here. This is what we just talked about. We got to talk about blah, 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 2 through 6, okay? All right. So this is divided up into several stages as well. Now, what I've been telling you guys, and maybe because I haven't been there, I haven't told you about the tattoos, but I always tell my kids that you could get some biology terms tattooed on you. That would be fantastic. One of them is the stages of mitosis. And if we were to break them down using their first letters, it stands for PMAT. Okay, PMAT, 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 PMAT. Don't forget that, PMAT. And if you want to, write that beside mitosis, okay? Good old PMAT, okay? All right, so let's talk a little bit about prophase. The P stands for prophase, okay? Now, prophase, we're going to have three things happening, all right? And actually, I hate that I'm not there to do this with you guys, but I want you to compare this diagram to this diagram, okay? Although that one's kind of weird, all right? What are some things that you see happening here? All right. One thing you should notice is that the nucleus looks like it's dotted, okay? And thank God that's the first one that I have up there. So the membrane of the nucleus is actually going to start disappearing. All right. It's a wacky phenomenon, and it's one that I have a hard time wrapping my head around. This is actually happening inside of us right now as we speak. Okay. Really weird stuff, but the nucleus can actually disappear for a little moment in time. So basically, our DNA is just free out there. Okay. So the membrane of the nucleus is going to disappear. Okay, membrane, nucleus disappear. That's the first thing. The second thing, if you notice, you can kind of see our X's here, okay? And we didn't have any DNA in the other one, but here you can actually see the chromosomes. So what's happening with B is that chromatin, that loose mass of DNA, that relaxed form of DNA, it's actually going to wind up into chromosomes. Okay, so it's going to spool around those histones, spool, okay, until it becomes that X. All right, and that's basically doing that to make it easier to move the DNA. If we tried to move 6 feet of DNA, all these guys right here are little pieces of DNA. Okay, if we were tried to move all of those without winding them up, it would be a nightmare. Okay, it's kind of like when you wash your car and the hose gets trapped on something. Okay, it's better to have one of those little spool things where the hose is on to drag it from point A to point B than to keep getting the hose trapped under your car. Does that make any sense? Okay, it's kind of a nightmare. All right, so the chromatin is going to wind up into chromosomes so we can see it. Okay, now the very last one you can't see in this picture, but in the picture that you guys are going to draw here in a second, you can see them. Just simply put down what's in black. You don't have to put down the red. The spindle fibers are going to appear. Okay, spindle fibers are kind of like fishing line. All right, so spindle fibers are going to appear, and you're going to draw it on this one over here once we get done with these notes. Okay, the spindle fibers are going to appear. Okay. All right, so that is going to be our prophase. Okay, that is our prophase. So let's do metaphase, and then we're going to take a break. Okay, so P is prophase. Now we have PMAT. So what's going to come next is M. So what M stands for is, whoops, back up before we go to M. Let me show you a cell in prophase. Okay, so if you guys look right here, this is my plant cell. This was our interphase. You see it right here, okay, where you can see the nucleolus, you can see the nucleus. Here, there is no nucleus. It's just kind of stuff floating around. Now, you can have different stages of prophase. This is prophase, just basically getting the party started. And if you notice, right here, you can kind of see the DNA a little bit better. Those squiggly things are actually going to be our chromosomes. And then right here, they kind of look like worms, okay? Those are the actual chromosomes. No, you can't see the X's, but you can get an idea of the squiggles, okay? Notice all the interphases around there and only 2 prophases, okay? Kind of an interesting observation. All right, in us, not that exciting. Plants are way cooler. Right here, you can actually start seeing a little bit more. This right here would be an example of interphase. Here, you kind of see something going on, okay? All right, let's talk about the M, which is metaphase. All right, so here's this picture. Everybody stare at that one. Compare it to this picture. Okay, what's going from this picture? What's going on from this picture to this picture? Okay, probably one thing that you should notice is that the chromosomes are lined up in the equator or the middle of the cell. Okay, these lines right here are the spindle fibers. Okay, you can actually see those. So what's happening here is that the spindle fibers are going to attach to the centromeres and pull the chromosomes to the middle of the cell. Meta, middle, middle, middle. Okay, meta, middle. That's what I always tell my kids. Okay, so the spindle fibers attach to the centromeres and pull the chromosomes to the middle of the cell. Meta, middle. Okay, so the suckers are lined up right smack dab in the middle. Pretty cool stuff. Okay. All right, so once you guys are done writing that, Miss Hartsock, why don't you hit pause on this video, give them about a 2-minute stretch break, okay? And then we'll hop back to this exciting stuff, okay? Pause. All right, guys. So I kind of prematurely paused because I always forget that I have these cell pictures afterwards. But basically, notice again, metaphase lined up in the middle, okay? So, ooh, this is cool. This is an actual picture of, and I don't know what organism we're looking at here, but all that blue stuff is actually the chromosomes. All this green stuff is actually going to be the spindle fibers. I think that looks so cool, okay? But again, I'm a big dork, okay? If we were to go back to our plant cell, all right, again, all of these are interphases. Here's your prophase. That looks like an early prophase, a late prophase. Right here, if you notice, it's kind of a blob right smack dab in the middle. It's not as spread out as this one right up here. So this is going to be your metaphase. And what's cool about this one is you can actually see the spindle fibers on that, which is really cool. Okay, so blob right smack dab in the middle. When you get to us, ours looks awesome here, okay? So what you notice is a straight line. That's just a blur of chromosomes lined up in the center. This one, you can actually see the spindle fibers as well. Very cool. Here looks like a prophase. There looks like an interphase, okay? So again, we're kind of seeing them blah together, okay? So PMAT, P is prophase, M is metaphase, A is my most absolute favorite, and I don't know why. I think I just like watching them being ripped apart, okay? A stands for anaphase, okay? So A is anaphase, all right? So let's compare this picture to this picture, okay? So stare at that picture, stare at this picture, what looks like is happening, okay? So what's happening here is that we're actually pulling apart those X's, okay? So this is where we perform our surgery to separate our Siamese twins, our sister chromatids, okay? Which are identical copies of each other. What we're doing is that we are separating them and pulling them to the opposite ends of the cell. Now how this happens is a pretty complex process, okay? We're not going to get into it, but I kind of like comparing this to going fishing. Whenever you throw those spindle fibers out there, what they want to do is they want to catch a chromosome, kind of like you casting your rod into a lake and casting your line into a lake and you want to catch a fish. When you catch a fish, you reel it in, right? Think of this as doing the same thing. They caught a, quote, fish or a chromosome, both sides, and they're reeling it in, okay? And we separate them. All right, so the sister chromatids are separated and pulled to the opposite ends of the cell. All right, this one looks so cool under a microscope. Like I said, it's my favorite. Okay, so they're being pulled to opposite ends of the cell. Okay. All right, so let's look at some real pictures of this. Okay, right here. Oh my God, that looks so cool. All right, so again, note surrounding this, all interphases. Okay, again, how do I know? You can actually see that nucleolus. Nothing fancy going on. I can't see chromosomes. This guy right here, if you notice, it looks like things are being pulled apart, okay? It kind of looks like fingers being drawn apart from each other. It's really weird, all right? And then there's the spindle fibers. They say spindle apparatus. It's basically the spindle fibers. Really cool. Okay, if we were to look at this one in our cell, again, pretty darn cool. What you see is basically things being ripped apart. Okay, it looks a little bit different than the diagram that I gave you. Well, just because that's a diagram and this is the real deal. Okay, so that is going to be your anaphase. All right, so P, prophase, M, metaphase, A, anaphase, T stands for telophase. Okay, so we have several things going on here. Now, if you remember back in prophase, they had early and late prophase. I'm going to give you late telophase, the very last thing that you see in telophase, okay? Oftentimes, I refer to this very inappropriately as the booty phase because it looks like a tushy, looks like a buns, okay? So this is the booty phase, all right? And this is late telophase. Now, if you guys notice, probably the first thing that you notice is that the cell is starting to pinch in, okay? So, oops, I didn't do that one first. And this one you can't actually see in this diagram, but you will in the diagram that you're going to draw over here, okay? Basically, telophase is the opposite of prophase, okay? And if you guys want to write that somewhere, that would be pretty cool. You can simply say opposite of prophase, okay? So if you got prophase, you got telophase. So the nucleus is going to reappear. All right, again, you can't see it in this diagram right here, but you will over here. So the nucleus is going to start showing up again. Okay, B, my DNA is going to start unwinding. Okay, so I'm going to go from a chromosome, all right, back to that chromatin. The chromosomes are kind of like, hey, and it's a chromatid as well, okay? So it's a chromosome or a chromatid, same thing. All right, so this guy right here basically says, you know what, I'm back where I need to be. I can put on my comfy clothes. I don't need to have my fancy school clothes or my teacher clothes, okay? I can change back up into something comfy. So they're going to start unwinding, okay? So chromosomes are going to become chromatin, all right? And last but not least, one other thing is that the spindle fibers are gone, okay? They've done their job. They've went fishing, they separated them, now they can go away, okay? So the spindle fibers disappear. Now, one thing that I hope I add up here is that the cell's going to start to pinch in, okay? I didn't, okay? The cell's going to start to pinch in, which is very obvious. Again, we call it the booty phase, okay? All right, if we were going to look at this, if we were to look at the plant cells don't necessarily look like a booty, okay? And the reason why it doesn't look like a butt is because of the cell wall, okay? They're not very flexible. All right, they're not flexible at all. So what they actually end up getting instead of the booty phase is they get this line right here called the cell plate, okay, which is actually going to be that new cell wall that's going to separate those two new cells, okay? But if you notice, this looks very different from anaphase because it doesn't quite look like fingers. It looks more like blobs that are still kind of connected to each other, okay, but are eventually going to be separated from each other, okay? And the animal cell, there you go. There's your booty. Booty. One cheek, two cheek. Okay. We can look like a booty because our membranes are flexible. Okay. So it's starting to pinch in right here and right here. And you notice you still can kind of see the chromosomes. They're going to start unwinding. Okay. So again, telophase. All right. Good stuff. Good stuff. All right. Let's go over our very last one. Let's put a number 3 by cytokinesis, please. Put a number 3 by cytokinesis. Okay? So cytokinesis is the very last stage. The first one was interphase. The second one was mitosis. And now we have cytokinesis. Okay? Cyto means cell. Kinesis means cut. Okay? So it's going to be the division of the cytoplasm and the organelles. So everything that we made in interphase, basically we're going to ship off where it needs to be. And we're going to divide that stuff. Okay, and we're going to produce 2 identical cells. This is huge, okay? Once you guys write that down, put a big square around it. We're going to make 2 very identical cells. There's nothing that's different between them. Produce 2 identical cells, okay? Pretty cool stuff. They are identical copies of each other, and thank goodness that they are, okay? All right, good stuff, good stuff. Yeah, Madeline thinks it's good stuff too. I gotta roar. All right. So, those kind of those look good. All right. So I have some animations to show you guys really quick. Okay. Couple of things. All right. To see. Let's do this step through here. Okay. I don't really want to hear them talk. So DNA replication. We get the X's going on right there. Everybody make sure that you're looking. Okay. And there's some other steps that I didn't include. All right. Play, come on. We didn't talk about a centrosome, which is okay. I'm sure you guys are heartbroken. Okay. All right, so there's my chromatin. All right, it's the loose mass right there, and it's going to wind up into our chromosomes. Okay. Pretty cool stuff. You're going to see okay, there we go, winding up. You see the nucleus disappear. You see the spindle fibers moving. Okay. All right, so those spindle fibers are going to attach to the centromeres and drag those chromosomes to the middle in metaphase. Okay, meta-middle. Anaphase, you're going to see them get pulled apart. Okay, super cool. All right. After that, what you're going to get is that it's going to dent in. And for telophase, we're going to get our booty phase. These guys are going to unwind. Nucleus reappears, okay? And this is linked to this PowerPoint, so if you guys need to watch it again, you're more than welcome to. Ms. Hartsock, if they decide that they want to watch it again, you can play it for them again, okay? And then that's that, okay? Now, what I'm about, yes, I am going to play you Mr. W's mitosis rap. Oh, yeah. Okay, this song is pretty corny, but you know what? Mr. W, he's not bad, all right? He's not bad, so just listening to this, he's going to get into some things that we didn't talk about, but at the same time, it's kind of catchy. All right, so here we go. You're about to get rocked.
Guys, I want everyone's attention.
Oh, yeah.
We're going to go over what we did yesterday. We're going to go over the stages of mitosis.
Yes, I'm really showing you this, okay?
All right, everybody ready? All right, let's go. Interphase of cell division's longest part. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing, chromosomes get duplicated, DNA gets replicated, chromosomes are spread out so they can't be seen distinctly. But note the nucleolus, the ribosome factory outside the nucleus, the two centrosomes. They later make a spindle which will pull apart the chromosomes. Prophase follows, the chromosomes condense, each is made of two sister chromatids like an X. Each sister is a clone, the closest of kin, and a centromere connects them like Siamese twins. The nucleolus disappears and melts away as the cell takes a ribosome production holiday. The centrosome separates, starts spindle formation for separating chromatids and cell elongation. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In late prophase, pro-metaphase, the nuclear membrane disintegrates. The centrosomes migrate to the cell's opposing sides, and between them the fibers of the spindle wend and wind. The spindle's made of microtubule fibers which attach to chromosomes. A kinetochore is a protein patch that serves like a handle that the fibers can grasp. When they pull apart the chromosomes, splitting them in half, the spindle moves the chromosomes with nudges so fine into linear formation on the 50-yard line, a location equatorial, defined in metaphase, where the chromosomes are lined up in that middle place. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. The spindle fibers pull on the kinetochores. A cellular, molecular, mitotic tug-of-war. The centromere snaps, sisters get separated. Now these chromatids are chromosomes. They've been upgraded. The snap and separation defines anaphase. The A for apartness, for moving different ways. Kinetochore, spindle fibers separate. The sisters see I'm waving goodbye, calling out I'm gonna miss you. And the other spindle fibers push and grapple like felons, make the cell elliptical like a watermelon. In telophase, membranes form around the chromosomes that spread out as a nucleoli come on home. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In animal cells, there's a ring of microfilaments that form at the equator, and they cinch themselves in tighter, 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 tighter till the cell is in two pieces. Yeah, in animals, that's cytokinesis, but it's different in plants, and then the cell divides by building a new cell wall from the inside as the Golgi sends vesicles with cellulosa goo, which makes a plate, then a wall divides the cell in two. And instead of one mother cell, we now have daughters too. Identical twins, kinda old but kinda new. From your single cell beginning, this is how you grew. And for single celled eukaryotes, it's reproductive too. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. Mitosis, chromosomal ride in.
I would so be his friend. Just saying, you guys. Me and Mr. W, I think we would get along. Okay? Hopefully it wasn't too painful. I think he's pretty awesome. Okay, so let's look at, I just got two more. These are pretty quick. I'm going to show you this actually happening in real time. Okay?
Differential interference.
Shut up. We don't want to hear her talk. Okay, so what you got right here is, hopefully, it's going to start moving. You got your chromosomes. Okay? Notice that it was a blob. You're going to see them line up, get all together here. This guy will zoom in here. This is going to be metaphase. Okay? Watch anaphase. Anaphase is so cool. All right? Yeah. So you're going to see this side go this way and boop. Okay? And you're going to see them turn back to a goo. All right? And that's going to be telophase, and you'll see kind of a booty pinching in. All right? So there's still chromosomes. There's the goo. They uncoil. And then the booty phase, and you have two identical cells. That is so cool. You want to see it again? I do. I don't care if you don't want to, because I do. All right, so again, our chromosomes right here. This is basically showing mitosis. They're moving. They're going to start saying, hey, we need to line up. You can't see the spindle fibers because they're so fine, okay, in this particular one. All right, lining up in the middle, lining up, okay, lining up. All right, and then they're going to be pulled apart. Whoop, my favorite. God, I love anaphase. I don't know why. All right, then got the daughter nuclei there. Then they're going to turn to goo. Going to start pinching in two identical cells. Pretty darn cool. Okay. All right, one more. This is showing a bunch of cells doing it at once. This one's pretty quick, 19 seconds, okay? So all these cells are going to do it at once, which is pretty cool. All right. So then there's anaphase, there's metaphase. You're going to see them pulled apart. Anaphase, plow. Look at that, doubled all the cells. Pretty freaking cool. Yes, we're going to watch that one again. This one you can't see the chromosomes too well until they actually get to metaphase. Okay. So it looks like the nucleus disappeared there. They're getting to the middle. They're being pulled apart. Pow. Dang, that's cool. All right, last one I'm going to show you is any Big Bang people in the house. I think that some of you guys are. Little did you know that in the intro there, they actually show one phase of the cell cycle, okay? They're actually going to show a phase, so let's see if you guys know what it is.
Our whole universe was in a hot, dense state that nearly 14 billion years ago expansion started. Wait, the earth began to...
There it was. Did you see it?
Started. Wait, the earth began to cool the autotrophs.
What was it? Anybody know? One more time.
That nearly 14 billion years ago expansion started. Wait.
Pow. Oh, it's so beautiful. It's my favorite. What is it? Yes, it is anaphase. Good job. So next time you're watching that with your parents, you can be like, you know what, Mom, Dad? That right there's anaphase. And they're going to be like, what? You are so smart. And you're going to be like, yeah, I know. All right. So that's part of your homework to do. Okay, what is this? All right, so you guys are about to do a activity called yarn mitosis. Actually, before you do that, Ms. Hartsock's going to hand out a diagram that I don't have up here anymore. And you guys are going to draw in these little circles what's going on. I gave you colors. Actually, if you make some of the X's, I have like 3 X's in there, or X's, listen to me, chromosomes. If you make those different colors, that kind of makes it a little bit fun. You're more than welcome to do that. I also have labels. Make sure that you include the labels, okay? Do not take that sheet, leave it in the basket, okay? Do not take that sheet, leave it in the basket, okay? So that's going to be the first thing you're going to do. The second thing you're going to do is going to be this one. It's cell cycle yarn modeling. This is going to be a test to see if you guys can follow directions, okay? Dun, dun, dun. So what you have right here are basically telling you what to do step by step. There's actually pieces of papers, labels, that have chromosomes, chromatin, nucleus, and cell membrane. You need to put those things in the right location, okay? So you're going to make this, draw it, make sure that you label each of those pictures, okay? Make it, draw it, label, make it, draw it, label. And I think I asked you guys to do this a couple of times, or asked Ms. Hartsock to ask you guys to do it a couple of times, okay? What I'd like for you guys to do is to actually do it without reading these directions, okay? Be able to show me, if I were to say, show me prophase, that you could use the yarn to show me prophase, okay? That's pretty big, and it's hard work, so hopefully you're good to go with that, okay? You have some homework tonight. The homework is this guy right here. I really like this because it's kind of basically saying everything that I just said. It's so easy. Basically, the answers to all these questions are right here, okay? You don't even need your notes for this. This is just kind of reviewing everything that I talked about. It looks like it's really hard, but once you guys start going on it, you're going to be like, holy crap, this is easy, okay? You just got to be able to read to give me the answers, okay? All right, so make sure you get your work done, turn things in, and yeah, that's that, okay? Yeah, I'll talk to you guys later, okay? Bye-bye.